So when we learned about the convergence in crosstalk among significant signaling pathways, we have discussed two distinct types of cell surface receptors in this chapter, like G protein coupled receptors and receptor tyrosine kinase. Another type of cell surface receptor that is capable of signal transduction was discussed in chapter 11, namely integrins. Although these three types of receptors may bind to very different signals ligands, all of them can lead to the formation of phosphotyrosine docking sites for the SH2 domain of the adaptive protein GRB2 in close proximity to the plasma membrane. The recruitment of the GRB2 SOS complex results in the activation of RAS and transmission of signals down the MAP kinase pathway. As a result of this convergence, signals from diverse receptors can lead to the transcription and translation of a similar set of growth promoting genes in each target cell. Then divergence will be happening. Evidence of signal divergence has been evident in virtually all of the examples of signal transduction that have been described in this chapter. A quick look illustrates how a single stimulus, a ligand binding to GPCR or an insulin receptor, sends signals out along a variety of different pathways can be seen, and crosstalk is happening. Number of signaling pathways as if each operated as an independent linear chain of events. In fact, the formation of circuits that operate in cells are more likely to resemble an interconnected web in which components produced in one pathway can participate in events occurring in other pathways. The more that is learned from information signaling in cells, the more crosstalk between signaling pathways that is discovered. Rather than attempting to catalog the way that information can be passed back and forth within the cell, we will look at an example involving CAMP that illustrates the importance of this type of crosstalk. Cyclic AMP was depicted earlier as an initiator of a reaction cascade leading to glucose mobilization. However, CAMP can also inhibit the growth of a variety of cells, including fibroblasts and fat cells, by blocking signal transmitted through the AMP-MAP kinase cascade. Cyclic AMP is thought to accomplish this by activating PKA, the CAMP-dependent kinase, which can phosphorylate and inhibit RAF, the protein that leads the MAP kinase cascade. These two pathways also intersect at another important signaling effector, the transcription factor CRUB. CRUB, as described, and a terminal effector of CAMP-mediated pathway. It was assumed for years that CRUB could only be phosphorylated by the CAMP-dependent kinase or PKA. It has since become apparent that CREB is a substrate of a much wider range of kinase. For example, one of the kinase that phosphorylates CRUB is RSK2, which is activated as a result of phosphorylation by MAP kinase. In fact, both PKA and RSK2 phosphorylate CREB on precisely the same amino acid residue, SER133, which should endow the transcription factor with the same potential in each pathways. When we look at the structures, signals transmitted from a G protein coupled receptor, an integrin, and a receptor tyrosine kinase all converge in RAS and then are transmitted along the MAP kinase cascade. So extracellular matrix, neurotransmitters, hormones, and growth factors are there. Growth factors like EGF and PDGF will be there, and they just bind to the G protein coupled receptors and receptor tyrosine kinase, and just integrin is also there. So GRB2 and SOS, GRB2 SOS, GRB2 SOS will be there, and they just lead to RAS, and then RAS leads to MAP kinase cascade. Then we just closely look at the structure of these proteins. We can see that EGF receptor is there, growth factors associated with that. Then SH2 domain, GRB2 domain is activated, SOS will be there, and RAS GDP will be there, and that will like epinephrine is there, beta adrenergic receptor that binds to creates ATP, PKE will be there, RAF will be there, MAP MAP kinase, RAS2 kinase, and PKE will be there, and CREP transcription factor is there, and gene activity will be happening. An example of the crosstalk between two major signaling, that is what we have explained now. And cyclic AMP acts in some cells by means of the CAMP dependent kinase PKA to block the transmission of signals from RAS to RAF, which inhibits the activation of the MAP kinase cascade. In addition, both PKA and the kinase of the MAP kinase cascade phosphorylate the transcription factor CREB on the same serine residue, activating the transcription factor and allowing it to bind to specific sites on the DNA. A major unanswered question is raised by these examples of convergence, divergence, and crosstalk. How are the stimuli able to evoke distinct responses even though they utilize different pathways? PI3K, for example, is an enzyme that is activated by a remarkable variety of stimuli, including cell adhesion to the ECM, insulin, and EGF. How is it that activation of PI3K in an insulin stimulated liver cell promote GLUT4 translocation and protein synthesis, whereas activation of PI3K in an adherence epithelial cell promotes cell survival? Ultimately, this contrasting cellular response must be due to differences in the protein composition of different cell types. Part of the answer probably lies in the fact that different cells have different isoforms of these various proteins, including PI3K. Some of the isoforms are encoded by different but related genes, where others are generated by alternate splicing or other mechanisms. Different isoforms of PA3K, PKB, or PLC, for example, may bind to different sets of upstream and downstream components, which could allow similar pathways to evoke distinct responses. This variation in response elicited by different cells possessing similar signaling proteins may also be particularly explained by the presence of different protein scaffold in each of the cell types. The specificity of a response can be orchestrated by the scaffolds with which the signaling proteins can interact, but it isn't likely that variations in isoforms and scaffolds can fully explain the extraordinary diversity of cellular response any more than differences in the structure of neurons can explain the range of responses evoked by the nervous system. Hopefully, as the signaling pathways of more and more cells are described, we will gain a better understanding of the specificity that can be achieved through the use of similar signaling molecules. The next topic is the role of NO, or nitric oxide, as an intracellular messenger. During the 1990s, a new type of messenger was discovered that was neither an organic compound such as AMP nor an ion such as A++. It is an inorganic gas, nitric oxide. NO is unusual because it acts both as an extracellular messenger mediating intracellular communication and as a second messenger acting within the cell in which it is generated. NO is formed from the amino acid L-arginine in a reaction that requires oxygen and NADPH, and that is catalyzed by the enzyme nitric oxide synthetase. Since its discovery, it has become evident that NO is involved in a myriad of biological processes including anticoagulation, neurotransmission, smooth muscle relaxation, and visual perception. As with many other biological phenomena, the discovery that NO functions as a messenger molecule began with an accidental observation. It had been known for many years that acetylcholine acts in the body to relax the smooth muscle cells of blood vessels, but the response could not be duplicated in vitro. When a portion of the major blood vessels, such as the aorta, were incubated in physiologic concentrations of acetylcholine in vitro, the preparation usually showed little or no response. In late 1970s, Robert Fargot, a pharmacologist at a New York State Medical Center, was studying in vitro response of species of rabbit aorta to various agents. In his earlier studies, Foucault used stripes of aorta that had been dissected from the organ. For technical reasons, Foucault switched from stripes of aorta tissue to aortic rings and discovered that this new preparation responded to acetylcholine by undergoing relaxation. Further investigation revealed that the stripes had failed to display the relaxation response because the delicate endothelial layers that lines the aorta had been rubbed away during the dissection. The surprising finding suggested that the endothelial cells were somehow involved in the response by the adjacent muscle cells. The subsequent studies it was found that acetylcholine binds to receptors on the surface of endothelial cells, leading to the production and release of an agent that diffuses through the cell's plasma membrane and cause the muscle cells to relax. For technical reasons, then it was changed, the experiment was changed, and in consequent studies it was found that acetylcholine binds to receptors on the surface of endothelial cells, leading to the production and release of an agent that diffuses through the cell's plasma membrane and causes the muscle cells to relax. The diffusible relaxing agent was identified in 1986 as nitric oxide by Luis Ignarro and UCLA and Salvador Moncada at the Wellcome Research Labs in England. The steps in the acetylcholine-induced relaxation response was illustrated like a signal transduction pathway that operates the means of NO and cyclic GMP that leads to the dilation of blood vessels is there, like endothelial cells are there, ACH, Ca2 plus will be there, nitric oxide synthetase, lumen, and guanylyl cyclase, GTP, GAMP, CGMP, relaxation happening, and smooth muscle cells are relaxed. The binding of the acetylcholine to the outer surface of an endothelial cell signals a rise in cytosolic Ca2 plus concentration that activates nitric oxide synthetase. The NO formed in the endothelial cell diffuses across the plasma membrane and into the adjacent smooth muscle cells, where it binds and stimulates guanylyl cyclase, the enzyme that synthesizes cyclic GMP, which is an important second messenger similar in structure to CAMP. Cyclic GMP binds to a CGMP-independent protein kinase, which phosphorylates specific substrates, causing relaxation of the muscle cell and dilation of the blood vessel. Now NO as an activator of guanylyl cyclase. The delivery that NO acts as an activator of guanylyl cyclase is made in the late 1970s by Farid Murad and colleagues at the University of Virginia. Murad was worked with ASIDE like N3, a potent inhibitor of electron transport and change, to discover that the molecule stimulated cGMP production in cellular extracts. Murad and colleagues ultimately demonstrated that ASIDE was converted enzymatically into nitric oxide, which served as the actual guanylyl cyclase activator. The studies also explained the action of nitroglycerin, which had studies also explained the action of nitroglycerin, which had been used since the 1860s to treat the pain of angina that results from the inadequate flow of the blood to the heart. Nitroglycerin is metabolized to nitric oxide, which stimulates the relaxation of smooth muscles lining the blood vessels of the heart, increasing blood flow to the organ. The therapeutic benefits of nitroglycerin were discovered through the interesting observation. Persons with heart diseases who worked with nitroglycerin in Alfred Nobel's dynamite theory were found to suffer more from the pain of angina on days they were in at work. It is only fitting that the Nobel Prize, which is funded by a donation from Alfred Nobel's estate was awarded in 1998 for the discovery of NO as a signaling agent inhibiting phosphodiesterase. The discovery of NO as a second messenger has also led to the development of Viagra during sexual arousal. Nerve endings in the penis release NO, which cause relaxation of smooth muscle cells and lining of penile blood vessels and encouragement of the organ with blood. As described above, NO mediates this response in smooth muscle cells by activation of the enzyme guanylyl cyclase and subsequent production of cGMP. Viagra and related drugs has no effect on the release of NO or the activation of guanylyl cyclase, but instead acts as an inhibitor of cGMP phosphodiesterase, the enzyme that destroys cGMP. Inhibition of this enzyme leads to maintained elevated levels of cGMP, which promotes the development and maintenance of an erection. Viagra is quite specific for one particular isoform of cGMP phosphodiesterase PDE5, which is the version that acts in the penis. Another isoform of the enzyme PDE3 plays a key role in the regulation of heart muscle contraction, but unfortunately it is not inhibited by the drug. Viagra was discovered when a potential angina medication had unexpected side effects. Recent investigation has revealed that NO has a variety of actions within the body that do not involve production of cGMP. For example, NO is added to the acid group of certain cysteine residues in well over 100 proteins, including hemoglobin, RAS, iodine channels, and caspases. This post-translational modification, which is called as nitrosylation, alters the activity, turnover, or interactions of the protein.